Our first speaker is Eva Kisdi from the University of Helsinki, Finland, and she will talk about evolutionary doctrine of a magic trait. Please.
Yeah, thank you very much. Let me start with something everybody knows, that speciation, non-allopathic speciation, or let's say for simplicity, sympathetic speciation needs ecological divergence coupled with reproductive isolation. So classic models, I mean models in the 70s, 80s, modeled this situation by assuming that there are separate loci controlling an ecological trait and controlling a mating process, something like associative mating. And then in this case, we need an association between the ecological loci and the associative mating loci. So linkage disequilibrium is necessary to get non-allopathic speciation going, but unfortunately, this association is very effectively destroyed by recombination. So the conclusion was that speciation can happen only if natural selection is strong, recombination is low, and penetrance of associative mating is high. And of course, if we want to generalize a little bit on these latter two, then we really need to assume strong natural selection to counterbalance recombination. Now, contrasting to this classical picture, we have now what we call magic traits, traits which are ecological traits under natural selection, but at the same time they also control mating. So the ecological and the mating traits are one and the same. And this, by magic, sort of, solves the problem of recombination. If the same loci control for both natural selection and mating, then we won't have this problem of destroying the association. And if a magic trait diverges, like body size in sticklebacks diverges, then this divergence implies reproductive isolation, so to speak, for free. So it looks like we have solved the problem of sympathetic speciation, but in this talk I'd like to argue that the magic traits are actually not a free ticket to sympathetic speciation, and problems arise. It is true that divergence implies reproductive isolation, but divergence becomes more difficult in the first place because of sexual selection, which is counteracting natural selection when mating associativity also depends on this trait in question. And even if we get initial divergence, then we can run into a problem later. As the two branches of the evolutionary tree grow apart from each other, it is a question whether they will grow far enough such that associative mating can indeed produce strong reproductive isolation, strong enough for us to recognize the two populations as two separate species. Obviously, even if mating is associative, but the two branches, the two phenotypes are too close to each other, then females cannot distinguish between the males, and with the best wish on earth, they cannot mate associatively enough to cut gene flow between the diverging populations. Now, the first question, whether divergence occurs or not, can actually be addressed in a very general framework using the methods of adaptive dynamics, because whether or not divergence by evolutionary branching occur depends on the local properties of the fitness function and of the mating function. So this can be done analytically and in a general framework, and this will be the subject of the talk of Tadeusz Priklopil tomorrow. And let me also point out that what I'm talking about is also joint work with him. But for the second question, we need the global properties of the fitness function. And therefore, I have to resort to an example, which is going to be the Levy model. Again, something everybody knows. So we are assuming that there are two contrasting habitats. The offspring are randomly assigned to one or to the other habitat. There is a continuous phenotype or trait. This is the magic trait, such that the optimal phenotype in habitat 1 and in habitat 2 are different. After selection, we assume that fecundity was high enough that both habitats are saturated, and eventually a fixed fraction of the adult population is coming from the first habitat and the rest comes from the second habitat. And then mating is either random, or if we already introduce associativity, then these adults form a single mating population but mate associatively with respect to the magic trait. Now we chose this very simple example because its population genetics is extremely well known, and also its adaptive dynamics is well known under diploid, sexual reproduction, and random mating. So this is what happens. This is again the trait value. Now mating is still random. If we start with a single monomorphic population, then we can get evolutionary branching such that two distinct alleles develop in the population. We are talking about diploids, so two alleles mean that there are two homozygote phenotypes, but because we are also talking about random mating, this is of course not speciation. We just get a genetic polymorphism segregating in a randomly mating population, and we have the heterozygote phenotype in the middle. We assume additive genetics for the trait. So all the time after branching, we have three phenotypes corresponding to the two homozygotes in black and the heterozygotes in the middle in green. This sort of evolutionary branching happens in a fairly wide range of the parameter space. There are only two ecological parameters in this model: the difference between the habitats, so how strongly contrasting they are, and the relative size of the first and the other second habitat. And in this shaded part of the parameter space, evolutionary branching produces a polymorphic population, but under random mating still. Now, actually, in this talk, I will confine myself to equally sized habitats. So actually, I will only walk through this middle line here. So simply, if the two habitats are sufficiently different from each other, then we get polymorphism, and otherwise there is an allele, which is an evolutionary stable phenotype in a monomorphic population. Now, on this basis, we want to introduce a mate choice model, and here we will follow the assumptions of Döbeli and Gavril and Zborka. So we envisage a population where females encounter males at random, and once they have encountered the male, they look at the phenotype of the male, and they accept the male with a certain probability, pi, and this probability is a function of the difference between the trait of the male and the trait of the female, and the probability of accepting the male is falling away from 1 to 0 as the absolute value of this difference is increasing. And how fast the probability of accepting decreases with the difference in the trait value is how associative mating is. If this function is very narrow, then the females are very choosy. If this is virtually a straight line, so a very broad Gaussian function, then the females don't actually care. So the inverse of the width of this Gaussian is what we might call associativity or preference or choosiness. There are several names in circulation. Now we will assume for simplicity again that females have infinitely many encounters, so they can try and try and try until they are mated. They are mated only once, so once they accept somebody, then it's end of the story, but they can keep trying forever. So there is no cost to mate choice to the females. Yet there is sexual selection from this scheme of associative mating. There is sexual selection against rare males. And this is something what Maria has already explained, but let me repeat it because this is indeed important in this model. For a female, the most likely thing to happen is to encounter a common male. For a common female, that's all right, they are happy together. But now for a rare female, who is different from the common male, she sees that this male is not really my type. There is a difference in the trait value. So I don't automatically accept this male, but there is a positive probability of accepting because there are mistakes in evaluation and everything like that. So the rare female might, say, reject the common male on the first encounter, but the second encounter is likely to be with the common male again, and she might accept or might not accept, and this story continues for a long time, and on average she has to reject a large number of common males before she can encounter her real sweetheart, the rare male. And in many of the cases, the rare female is mated by, if you like, by mistake, so by accepting the common male, because there is a positive probability of accepting the common male. So many rare females are already mated before they had a chance of encountering a rare male. And this means that the rare males don't get their matings. So there is selection against the rare males. Now this has consequences already on the short population genetical time scale, so on the time scale where numbers of individuals and frequencies of alleles change. First of all, because the sexual selection operates against the rare variants, the rare alleles, obviously it can cause the loss of polymorphism. But it's also important that sexual selection against the rare males is the strongest when there is indeed a big difference in frequency, one type is really common and the other type is really rare. If the types are about at the same frequency, then everybody has the same disadvantage. So then the differences in selection, sexual selection, are small. So sexual selection gets weaker as the types have more equal frequency. This means that we can have multiple stability. It could be that fixation of an allele is stable, because when we introduce a new rare type, then it is so much selected against through this sexual selection that the new allele cannot invade. But if the two alleles are about of the same frequency, they can still coexist. So we can have polymorphism, but this type of polymorphism is then not protected. When an allele becomes rare, either by an environmental disturbance or just by genetic drift, then it will not recover. It will go actually now deterministically to extinction. So to illustrate this, look at this figure. This comes from the Levy model with a very simple setup with fixed fitnesses within habitats. If we have zero assortativity random mating, then the model produces protected polymorphism as the Levy model should. If we have no natural selection, so the habitats are not different or the within habitat fitnesses are equal, only assortative mating, then we get fixation of one or the other allele, because always the commoner type will win. There is sexual selection against the rare type. And finally, if assortativity is high enough, but there is also natural selection, then we have this kind of unprotected polymorphism. When natural selection keeps the two alleles about equally common, then sexual selection is weak and there is a stable equilibrium. But should one allele get really rare, it cannot recover anymore. So the polymorphism is not protected. Now going to the next time scale, when we consider long-term evolution by repeated invasion and substitution of alleles. So now we go to the adaptive dynamic time scale of a magic trait. That is a trait which is now under natural selection according to the Levy model, and also controls mating according to this female mate choice model. Because sexual selection is stabilizing, the most common types are always preferred. This can inhibit evolutionary branching, but again, it can also cause multiple stability. So as we will see, it is possible that two separate species represent a stable equilibrium, but we cannot get to this equilibrium, so there is no speciation. Once we have the species, it's all right, but we can never get from a monomorphic population to two species. And finally, there is a different issue which I will treat only very briefly in this talk. We could also look at the adaptive dynamics of choosiness. So if we go back to this figure, the magic trait which I will look at in most of my talk is this trait phi, and the probability of accepting the mate depends on the difference in the magic trait. But assortativity, this sigma, is also a trait which might evolve. And this is actually very interesting because the evolution of sigma, the evolution of choosiness, shows how a trait becomes magic. If we start with a non-magic trait, with a wink to Harry Potter, one might say, if we start with a muggle trait, can it ever become magic? So can it evolve assortativeness based on itself? So, well, these authors have looked at the problem, and the conclusion is that choosiness can stop increasing at an intermediate level, so speciation can start up, but then it stalls and does not go to completion. And again, there is a multiple stability. It is possible that full isolation, the existence of two species is stable, but it is an equilibrium which is unreachable from random mating, so we can never get there. Speciation stalls. So in this talk, I will mostly focus on the second problem. And well, the first population genetic thing comes up as an intermediate step, and at the end, I will a little bit talk about the adaptive dynamics of choosiness as well. So going to the adaptive dynamics of a magic trait. First, I produce here the so-called pairwise invisibility plot. So I'm sure that many of you are familiar with this kind of representation of possible invasion events. On the horizontal axis of every plot, there is the resident allele. So the resident population is monomorphic for an allele which produces a certain phenotype represented on the horizontal axis, and the vertical axis is the mutant allele, and then these signs plus and minus show if the mutant can invade or cannot invade. In the first pairwise invisibility plot, we have still random mating, and under random mating we get evolutionary branching. From the plot, this you can see, I don't know if these faint little arrows are visible from the back, but they represent invasion and substitution events. So for example, starting from this resident, a mutant who has slightly larger trait value can invade because we are in the plus area. And once it invaded, it is a quite general theorem of adaptive dynamics that it will also go to fixation. So then we will have a new resident population with a slightly larger trait value, and then the story repeats, and we are going towards this so-called singular strategy or singular allele. But this singular allele is not an ESS because there is a range of mutants who can actually invade. And again, it is a general proof that at such a point, which is convergence stable, we are going there, but not evolutionary stable. We cannot stop there because further invasions are possible. This population will undergo evolutionary branching and will produce two distinctly different alleles. But because this is now under random mating, it is not speciation, just polymorphism. Now, if we do the same, but assume some positive assortativity, so now the females are choosy with respect to the magic trait, then what we see is that instead of a branching point, we get an ESS. So again, there is directional evolution towards the singularity, but once we arrive at the singularity, it is not invadable. All mutants now have negative invasion fitness, indicating that invasion is not possible, and this resident population with this generalist middle trait value is evolutionary stable. This is a final stop, and we are not getting anywhere towards speciation because we don't even getting polymorphism. And finally, if we really look at a high level of assortativity, then basically we still have an ESS, but still the shape of the pairwise invisibility plot changes, and the significance of that is shown here. These plots don't simply look at invisibility, but mutual invisibility. So can one allele invade the other and the other invade the first? So is there protected polymorphism? These black areas indicate those combinations of alleles where protected polymorphism is the outcome. So each allele invades the monomorphic population of the other, and we can conclude that these two alleles will coexist and form a polymorphism. Again, the first picture is for random mating, so here we just have two alleles segregating. And as we increase assortativity, a little gray area appears, which denotes unprotected polymorphism. So we go back now to this population genetic complications, that there is multiple stability, unprotected polymorphism because of sexual selection interacting with natural selection. And in this last figure, now there is a much wider area of unprotected polymorphism, and there appeared here a little white thing between the two lines where there is mutual exclusion. Whichever allele is there first is stable against the invasion of the other. So we will only see monomorphic populations, but which allele will be there depends on history, which was there first. Now these two plots indicate that it is more difficult to get polymorphic in the first place when we look at an associatively mating population relative to a randomly mating populations. So where we could get evolutionary branching and polymorphism under random mating, it is not possible anymore with assortativity. So because of stabilizing sexual selection, the prospects for speciation are diminished because of this. But what happens if we do get branching? How does the story continue, and will the two branches diverge far enough so that assortativity will provide strong enough reproductive isolation to consider the outcome to be two separate species? Now in these plots, first of all, the decorated areas again denote those allele combinations where we do have polymorphism. So in this area, the two alleles form a polymorphic population. Outside these areas, one or the other allele goes to fixation and the other goes extinct. Let's start again with random mating. And in this case, we still had evolutionary branching. Here in the middle, we have the branching point. And then these little arrows indicate the direction of evolution of the two alleles. Now, one resident allele is on the horizontal axis, and the other resident allele is on the vertical axis. And when the arrows point like this, to the left and upright, then it means that the higher, the allele with the higher allelic value will evolve to be even higher, so we are going up, and the allele with the smaller allelic value will evolve towards even smaller values to the left, so the two alleles are evolving away from each other, so it is branching. Near the branching point, we have divergence. But as we get away from the branching point, the orientation of these arrows can change in a complicated manner. And eventually, this evolving population will go to one or the other attractors in this plot, which are indicated with black dots here and here. And here in the middle, if I can point precisely enough, there is a saddle point, so then trajectories will go either to the left of the saddle and to the lower attractor, or right of the saddle and to the higher attractor. This is, of course, a symmetric situation. We break symmetry either in this way or in that way. But the bottom line is that there is a evolutionarily and convergent stable polymorphism, which will be the final outcome of evolution in this system. So there are two alleles such that their combination represents an evolutionary stable population where there is no more invasion. So these two black dots are the final outcomes of evolution. And under random mating, it can only be a polymorphism, obviously. Now let's increase assortativity. Assortativity is incrementally increasing between the plots. The first thing which happens is that instead of a saddle, we now see an attractor in a symmetric position. And if we do start from the branching point, so the initial population was monomorphic, and then it had to evolve to the branching point, and then we get the polymorphism, then this symmetric point will be the final outcome. But this symmetric point represents a population with two alleles which are pretty close to each other. So the two homozygote phenotypes are rather similar as compared to the width of the mating function. So the females cannot distinguish between the two types of homozygote males very well, which means that they cannot mate assortatively because there is not enough difference between the males to be clearly visible. So in this population, there is only very weak reproductive isolation. If we increase assortativity, then, well, the picture becomes more complicated. There are now three symmetric singularities of the allelic populations. And at this point, further away from the branching point, this larger black dot indicates that here reproductive isolation has got considerably stronger. Now, the F value familiar from basic population genetics is above 90%, meaning that more than 90% of heterozygotes are missing as compared to random mating. So this is still not enough to consider the two homozygotes to be two separate species, but at least it's something promising. The problem is that this nice big black dot is unreachable from the branching point, because if we start with a monomorphic population, evolve up to the branching point, and become polymorphic here, then evolution will stop at this smaller black dot, the first attractor as seen from the branching point, and at the first attractor, the difference between homozygote males is really very small, so assortativity is of no use, and the population is very close to Hardy-Weinberg equilibrium. And unfortunately, this singularity isolates the branching point from the almost speciation singularity further out. So yes, we could have two homozygotes which are almost separate species, but we can't reach it from a monomorphic population. And the situation is somewhat similar in the case of very strong assortativity. Here again, there is a diallelic singularity where really now so much of the heterozygotes, or if you like hybrids, are missing that we can declare that this point represents two separate species. The two homozygotes are reproductively isolated, but again, we can't reach it from the branching. Not only that we can't reach it, but by this time the branching point has turned into an ESS, and then there is a saddle here which isolates the ESS from the speciation singularity. So the two species are stable, but they are unreachable. Now, this was just one example illustrating the possible evolutionary scenarios with a moderate difference between the two habitats. Let me show another example where the difference between the two habitats is actually even smaller a little bit, but the conclusion is rather different. On the random mating, again, we just get polymorphism, but if we increase associativity, then suddenly there is no d-allelic singularity at all. So from any d-allelic population, the system evolves towards the monomorphic singularity. It's an ESS, a monomorphic ESS, and every polymorphic population will eventually go to this monomorphic ESS, so we will lose polymorphism. This third picture is just to illustrate how we can actually eventually get a speciation singularity with strong associativity, but again, this is unreachable. Now here, if we have two separate species to start with, but then there is a change in the environment which changes associativity. Now I have something like the famous cichlid example in mind, that the environment changes such that the cichlid females cannot recognize their corresponding males anymore because the water has become turbid and therefore the colors are not properly visible, so associativity is physically not possible. If there is a change like that, so suddenly there is a drop in associativity, then this same point corresponds to the point here, which means that now the two species not only mix and more or less mate randomly, but also they start to evolve towards a monomorphic ESS. So if we can quickly restore the original environment, then we can restore two species. But if it takes some time so that the species can evolve in the meanwhile, then they will get closer to the monomorphic ESS, and from that point on, there is no return. It will continue even if associativity is restored. Finally, we will lose polymorphism, and there is no way to get back to two species. Actually, the system from a mathematical point of view is very nicely complicated. There is a rich bifurcation structure with respect to the ecological parameters, so how different the two habitats are, and with respect to associativity. These two brown lines here show the cross sections, what I showed on the previous slides. But of course, what we would like to know is where is speciation. So now I overlaid the line where 99% of heterozygotes are missing. And below this line, there is a speciation singularity, but the speciation singularity is reachable only in this area. Whereas here, there are two species stable, but it is not reachable from the branching point, or the branching point doesn't exist. Another quick example, just one slide on what happens if the patch sizes are asymmetric. Then we can actually get an example where the speciation singularity is in the area of unprotected polymorphism. So it means that we can have two reproductively isolated species stable, but one of the species is not protected. The population dynamics is something like this. And if the first species goes extinct and the second species gets fixed, then there is no way that the first species could invade back, and the second species will evolve to the ESS. Now, one slide about the evolution of associativity. This is again the same what was my first example to walk you through this d-allelic evolution story. So this is a singularity where we have a stable polymorphic population but very weak reproductive isolation. But now we have a theorem from Pennings et al. Saying that if heterozygotes have lower fitness than homozygotes, then choosiness is always selected for. This is a very powerful theorem, and because at this symmetric point heterozygotes are indeed having lower fitness than homozygotes, we know that choosiness will evolve higher, so we can hop between these pictures and we can arrive at a speciation singularity. It is true that this is such a point where we do have separate species, but if one of them goes extinct, then there is no recovery. So this is not, maybe not such a safe situation for a very long time span, but still we have stable separate species. The problem with this picture is that the first hop we can't make. Where I started from was a population with weak associative mating. But if we start from random mating, then actually we go to an asymmetric attractor, and the symmetric position is occupied by a saddle. So here, some associativity must arise very quickly in order to get from the first random mating picture to the second one. So there is a relatively short period of time where associativity has to increase, or else we end up at a singularity where heterozygotes are not selected against, and therefore speciation cannot take off. So to summarize, sexual selection or magic traits cause a number of problems. First of all, the divergence may not even happen. We can have an ESS in place of a branching point. And actually, this is the problem what we see most often in realizations. But even if we have evolutionary branching, it could be that divergence stops short of speciation, so the two branches do not get far enough. And the third problem is that choosiness may not evolve high enough. And then in addition to this, we had this last problem of having a limited time for choosiness to start evolving. So there is a time window for the onset of speciation. Now, all these problems go away if we assume that natural selection is strong. So if the habitats are really strongly different from each other, then natural selection will overpower sexual selection, and whatever naughtiness sexual selection is doing will go away, because natural selection is simply stronger. And actually, strong natural selection also has this time window issue. So all the difficulties go away if we assume strong natural selection. But you see, with this, we are back to square one. Sympathetic speciation happens only if natural selection is strong, just like in the classic models, but for different reasons. So if I can have 2 more minutes, I'm not sure, then I'd like to have an appendix here. So why magic traits after all? Because it is a fact that in almost every case study where we do see ongoing sympathetic speciation and we suspect that they will end up with separate species, those are always based on magic traits. So in the end, in empiry, the conclusion is that magic traits are the key to speciation. But if again we need strong selection like in the classic models, then why do we see only magic traits? Well, one suggestion I'd like to make here is that classic speciation models, as they assume the separate ecological locus and mating locus, they set up everything the simplest way they could for obvious reasons. So they set up the mating process such that the mating locus was selectively neutral. But this is a degenerate situation. Selective neutrality is a knife-edge situation between stabilizing or something. So obviously, if we change the model a little bit, then this neutrality will go away. And indeed, if we make the mating model a little bit more realistic in these classical models, then sexual selection will show up just as it is showing up in magic trait models, and it will lead to fixation. If there is no natural selection on the mating locus, it will lose genetic variability altogether. And if it is monomorphic, then it can't be the basis of speciation. So my suggestion is that if the mating trait itself is a magic trait, then natural selection can help maintaining the genetic variation necessary for speciation. Maybe sexual selection is weak. Maybe the classic models were not too far from reality. So in reality, there is only weak sexual selection. Then only weak natural selection is sufficient to maintain genetic variation, but still it's already a magic trait. And then we could look at models where we try to find association between mating traits, which are actually magic traits, and with other ecological traits, which are not mating traits, which are muggle traits, non-magic traits, but then see how this linkage, disequilibrium, or association with muggle and magic traits promotes speciation. Thank you very much.
Yeah. Thank you. We received a suggestion during the break that those who will ask questions maybe stand up, so it's easier to see who is actually asking the question. So if you could do that, then that would be much appreciated. You have a question over there from Andrew N. The last one.
So we modeled
completely simple.
I'm wondering what happens when you add a bit of, say, space in hair factor or something like that involved. That change dynamics of model versus magic traits.
Yes, of course it will help, and especially it helps the time window issue. A little bit of space takes away this problem of having a limited time to initial associativity to evolve.
There are more questions to, yeah, there are questions in the
It good. So,
this problem with the asymmetric solutions are
Yeah.
so the end point from there that no juiciness can evolve. Do you find that you think that emerges more or less easily if the ecological setup is asymmetric? We had all this symmetric things here, but what is your gut feel?
Yes, that's a very good question, actually. And I'm not quite sure now of the answer. But it's also true that this asymmetric solution exists in only an interval of the strengths of natural selection. If natural selection is weak, then we don't have this problem, but of course then it's too weak anyway. And if natural selection is strong enough, then again we don't have this problem. But I will look up what about asymmetric patches.
Okay, so while we're changing computers, we will take one more question here.
To what extent is this asymmetric solution an artifact only one locus for the ecological trait? Change loci for the ecological trait?
This is a more complicated question than it sounds, because if you do the same thing with two loci, but letting each locus evolve a la adaptive dynamics with many small mutations, then one locus loses polymorphism, and the other locus does what this model does.